Thank you for joining I Am Possible, which is India's first future tech meets sustainability podcast. Today, I'm delighted and honored to have with me Dr. Srikanth Ramaswamy, who is a computational neuroscientist, a principal investigator at Newcastle University, a Marie Curie Fellow, and ex-group leader at the Blue Brain Project that's been working on digitally reconstructing the human brain. So, Srikanth, really appreciate you taking time and being part of the podcast. Our brain, which is a 3 pound, it's one of the most magnificent machine, if you might call it, in the entire universe. So. Why don't we start with the human brain? How does it give, I mean, the question of how it gives rise to intelligence or consciousness or reality, it's a little way off, but why don't we start with how does the brain gives rise to a five perception?
So yeah, thanks a lot, Eddie, for having me on your podcast, and yeah, it's a great pleasure to chat. So to start with, we still don't know a lot about the human brain. So fundamentally, um, the the experimental model that's being used at laboratories worldwide are rodents primarily, right? So uh, the by and large, the organization of the brain, um, despite the fact that it kind of scales up linearly from mouse to monkey to man, is that this organizational structure is remarkably conserved. So the idea is that by trying to understand a more tractable animal system such as the mouse brain, it would be possible to try and extend these findings to understand how the human brain also works. Of course, um, the rodent brain is orders of magnitude smaller than the human brain, but in terms of, for example, how certain brain regions are structured, like let's take the neocortex, for example. So neocortex is because it's the most newly evolved part of the mammalian brain. Um, and the parts of the neocortex, for example, that deal with these five senses that you mentioned, they're all pretty much remarkably conserved from mouse to monkey to man. But the sophistication of these regions, of course, is quite linearly, in that the mouse brain possibly cannot do as many things as the monkey brain, and the monkey brain cannot possibly encompass all that the human brain can do. However, the substrate is pretty much the same in terms of the kinds of neurons involved, in terms of the kinds of laminar structures of where these neurons are organized, and in terms of the different brain regions that are actually earmarked to carry out these sensory processing modalities. All this is pretty much conserved. But the nuance lies in the fact that the structure of neurons, for example, how complicated certain parts of the neuron called dendrites are that actually receive inputs from other parts of the brain, and how complex are parts of the neuron called axons that actually convey um, the information that the dendrites receive to other neurons in different brain regions. So this, of course, differs because we again know over decades of quantitative analysis that neurons in the mouse brain are much smaller than in the monkey brain, which are again a lot more smaller than in the human brain. So the size of these neurons is actually very important in determining how they process information. Because, as I said, dendrites, for example, right? So let's talk about certain neuron types called pyramidal cells in the brain. So pyramidal cells are possibly the most abundant cell types in the neocortex. So the reason they're called pyramidal cells is they have a pyramidal shaped cell body, the soma or the nucleus of where a lot of information processing is thought to happen in neurons, right? So they really are shaped like a pyramid. So the apex of the pyramid structure of pyramidal neurons is the site where certain processes called apical dendrites emerge from, right? It's kind of a bit like a fountain where you have these processes protruding out of the apex of the pyramid, and they actually protrude upwards all the way to the surface of the cortex. And by virtue of this architecture, of this ubiquitous architecture, pyramidal neurons can actually sense inputs from different neuron types that are all projecting onto their dendrites. So there are fundamentally two different cell types in the cortex. So excitatory neurons, of which pyramidal cells are a part, and inhibitory neurons. So the fundamental working principle of excitatory neurons is that they excite. So the reason of their existence, they sample inputs from other neurons, make some sense out of it, and then convey this information onto other neurons to fundamentally excite them or to increase their levels of activity. Whereas on the other hand, there are other neuron types called inhibitory neurons, also in the cortex, whose fundamental role is to gather inputs from all across the brain, but to convey this input to inhibit or to depress the activity of neurons that they project, right? So depending on the layer where these neurons are located in, their dendritic and axonal shapes, they could either be excitatory, which are about 80% of neurons in the neocortex, or inhibitory, which are about 20% of neurons in the neocortex. So the function of these 20% or so inhibitory neurons is still enigmatic. We don't understand what these neurons exactly do, from mouse to monkey to the human brain. It's all the same. It's kind of still skimming the surface. And one of the primordial roles of these inhibitory interneurons in the mammalian brain is that they could kind of act like a fire extinguisher or like a brake. So imagine your pyramidal neurons are like your Formula One racing cars that are running through a circuit unhindered, right? So their essence is speed. Their essence is to go as fast as possible. But we know that speed thrills but kills, right? So we don't want these neurons to be constantly firing or conveying information all the time. So they need to have some kind of a break, some kind of a fire extinguisher that tells them that, look, take a pause, take a break, because you've already conveyed enough. And now is the time for the rest of the brain to digest what information you've been conveying, right? So these inhibitory interneurons out there really function as a significant brake, like the reins on a horse, to really pull the horse to a stop and tell the horse that, look, stop, this is it. For now, let's try and digest what you've been doing. And then when you try and run again, let's take it from there and do this process all over again. So this whole process of the interaction between excitatory pyramidal cells and inhibitory interneurons, I mean, this is lifelong, right? This goes on forever and ever and ever again. And what differentiates this interaction between mouse to monkey to man is the fact that the structure of dendrites and axons could pretty much scale linearly as you move across the mammalian hierarchy. And given the fact that dendrites of pyramidal cells or inhibitory interneurons are larger from mouse to monkey to man, there's a larger surface area, right? So they could simply accommodate a lot more non-linear processes in their dendrites or axons which could convey very unique computational abilities. So we know that these structures are different and that they could pretty much scale linearly, but how and why and what these different nonlinearities on the structures of dendrites and axons could mean in terms of computational ability of these neurons is still something we're trying to understand.
There is so much that we do not know. You mentioned that we are just about understanding a fruit fly and a rodent model. And you also mentioned that it scales up linearly. If we kind of are able to completely understand a mouse brain and how it functions, maybe we might be able to understand a monkey brain and then a human brain, which is still quite far away. And you mentioned that there is 80% of excitatory and 20% of inhibitory neurons, the 20% of which we do not know anything of. So
I wouldn't say we do not know anything about. It's just that there's a lot more diversity in these interneurons, and we're still figuring out why there is this diversity and why it matters.
right. So why don't we start with what do we know about it? Because there are, I mean, many startups, many institutes, research agencies working on understanding the brain better, and there's Blue Brain project we will get to, which is working on an in silico model trying to reconstruct the entire human brain in a computer. What's the current status of understanding the human brain, as in, you know, with the scanning, mapping and reading technologies with the brain computer interfaces?
Okay, so there are two ways with which current knowledge could try and help us understand the structure and function of the human brain. So of course, we all know about functional magnetic resonance imaging, but what this method could tell us about the brain is slightly qualitative, right? Because what this method could tell us is how certain brain regions are active during a certain task, right? So you place a person under an MRI scanner and show this person, let's say, a bunch of images, right? Like, I mean, there was this whole story quite some years ago about grandmother neurons and Jennifer Aniston neurons, right? So
What's grandfather neuron and Jennifer Aniston neuron?
There was this whole study about how certain parts of the brain, primarily in the temporal cortex, which is part of the neocortex, but which is supposed to play a very definitive role in processing images, right? Like in trying to understand what a certain visual image conveys, right? So these friends and grandmother cells, that was a very interesting study. And then through this study, it was established that there were certain brain regions that lit up when the image of Jennifer Aniston was shown to these people, and there were certain other brain regions that lit up when images of their own family members, like their own grandmothers, were shown, right? So this then conclusively demonstrated that there are certain brain regions that are a lot more active uh, when provided a very specific visual cue, right? Like Jennifer Aniston or your grandmother. Um, so this study concluded um, that the brain, the visual part of the brain, is parcellated into very specific areas that could then try and make out something that could resemble people from your own family, or information, visual information of your own family, or on the other hand, visual information from something that you're familiar with, like your popular sitcom, like Friends, right? But this is still very qualitative. It's kind of the tip of the iceberg. because this tells us what brain regions could be involved in processing these visual cues. But it doesn't quite tell us what are the different neuron types that are interacting to bring about this specific form of computation, right? So, a lot more recently, there's been a renewed interest in using human tissue samples to try and understand or to try and map out the diversity of the shapes of these neurons, their functions, how they interact, so on and so forth, to try and build a bridge between what's happening at the local level, at the local organizational level, and then link this to what could be happening at a more global network level, right? So to try and understand what's possibly happening at the local organizational level, over the years, neuroscientists have tried to harness resected human brain tissue samples that are otherwise discarded, that are of possibly not a lot of use to neurosurgeons who actually have to traverse the depth of cortical tissue to try and reach the epicenter of epileptiform activity in patients who are suffering from chronic epilepsy, right? To try and introduce certain deep brain stimulation paradigms to go to reach the root of epileptiform activity, which is more often than not centered in the so-called temporal cortex in the human brain. So the temporal cortex is quite a deep structure within the neocortex. And to get to the temporal cortex, where, or rather, which is thought to be the nucleus of epileptiform activity, you really need to go around a lot of healthy human brain tissue, right? And so all this healthy human brain tissue would have otherwise been discarded. And so over the past few decades, we have ways to try and use this piece of dissected brain tissue that people would then use in the lab to perform electrophysiological experiments to try and understand how the human brain is organized, the structure and function of these neurons. Really poke electrodes into neurons in human brain tissue samples, like inject a certain stimulus into them, an electrical pulse, to try and understand how these neurons respond, and to also look at how these neurons are connected, so on and so forth. That would then allow us to ad hoc understand the complicated structure, the dendritic and axonal structures of these neurons, and then compare them against mouse or monkey neurons in the same brain region. So this has then allowed us to understand how human neurons could be a lot more sophisticated in terms of their computational principles compared to mouse or monkey neurons. So this is really the window in trying to understand how human neurons could actually function. And very recently, like the past couple of years or so, we've also been able to insert certain sophisticated probes, electrical probes called neuropixels, to try and record electrical activity from epileptic human patients, to also, again, try and decipher how human neurons are so distinct and idiosyncratic, if at all, in terms of their function compared to mouse and monkey neurons. So this is where the state of the art is. So in a nutshell, it's very difficult to access human tissue samples, and these primarily come through means of dissected tissue from epileptic patients. So therefore, the bridge between what could be happening at the local neuronal level and the global network level in the human brain is still something that has a long way to go, in my opinion.
Right. You mentioned that the learnings that we have, the state of the art learning, is with understanding human tissue samples of the brain. And then you said there is ways to insert neuropixels and understand the language of the brain. Would you like to address the brain-computer interface? There's companies such as Neuralink and Kernel who are creating these brain-computer devices to kind of and then there's DARPA who's working on the N3 initiative, which is the non-invasive BCIs. Would that be eventually a way to kind of understanding the language of the brain, because we've got around 70 billion neurons, 100 trillion synapses, and the way they fires gives rise to all of our senses. But you said we're still kind of far away from understanding the basic language of the brain. You being a computational neuroscientist, how is this going to come together? What is your foresight on this, and address the brain-computer interface also?
Okay, there's also an important missing link that I haven't touched upon yet. The so-called wiring diagram of the brain, right? The so-called architecture, the blueprint. So to build a house, you would typically go to an architect, right? Who would then tell you what you can do in this little plot of land that you have. Right? So maybe you want to build a little house, maybe you want to build a multi-storied house, whatever, right? So principally, you need a blueprint to try and understand how you build a house, or in terms of the brain, how you try to understand how the brain is organized, right? So the blueprint of the brain is the so-called wiring diagram. And this wiring diagram is not carved in stone. This wiring diagram is something that dynamically evolves. That's evolving as we speak in this podcast, right? And what's causing this wiring diagram to evolve? So there are certain chemical messengers in the brain called neuromodulators, right? So you might have heard of certain chemical signaling messengers like acetylcholine or dopamine. Dopamine, it's called the pleasure molecule and things like that. So there are the big five neuromodulatory systems, chemical signaling messengers in the brain. So I like to call these through the mnemonic hands. So H for histamine, A for acetylcholine, N for noradrenaline, D for dopamine, and S for serotonin. So these are really the big five neuromodulatory systems in the brain that exploit the wiring diagram of the brain, right, to then guide it to reach to a certain state called a brain state, right? So if the wiring diagram is kind of like a sponge cake that you typically find in a bakery, what these neuromodulators could do is really to provide the icing on the cake to really make the cake look very different. So how these neuromodulators are structured and how their function is very fundamental to understand brain function. And we're only now beginning to decipher how these neuromodulatory systems project, for example, to the neocortex, and how they function on the different neuron types in the neocortex to bring about changes in brain states. And the sophisticated thing about these neuromodulators is that they can operate on a so-called bimodal manner, where there are all these neuromodulatory projections, which are actually axons from these neuromodulatory neurons that actually have a very huge surface area of projection. And when they get to their target, they could really spritz or spray a certain neuromodulator that could then target all neurons around the vicinity of this axon, or they could also explicitly, physically contact certain neurons in their sphere of influence, right? Um, and this is a very complex problem because depending on what neuromodulatory axon is projecting and what neuron type is in the vicinity, there can be very differential effects because in some way it's like a lock and key problem because it's a certain key that can open a lock, and if the lock are neurons in the cortex, the key is really the function of these neuromodulators. Um, and these neuromodulators don't function in isolation. They always interact. It's kind of like a tug of war competition because one neuromodulator sets the stage for another to take over. So it's kind of a relay race that's all happening in parallel as we speak. And so this is a very complex problem. And to understand how these neuromodulators are organized, how they function on different neocortical neuron types, uh, ultimately bringing about changes in brain states and behavior is a very grand challenge in modern neuroscience. And we are only now beginning to come up with the kind of tools to understand how these neuromodulators work. Uh, but this is again only in the rodent brain, maybe partly in the macaque brain or the monkey brain, but we're still not there yet to try and understand what's happening in the human brain. So this is still a very complex problem. And the, and neuromodulatory function is really fundamental to try and understand the structure and function of the brain because without neuromodulators the brain wouldn't possibly do anything in my opinion.
Right, right. So you have been part of the Blue Brain project, which by founded by Henry Markram with a huge moonshot of in silico, building the brain in the computer. You were the group leader of Blue Brain project. Would you like to talk about the vision itself? Where are you guys at this point in time? And your role when you were there with the Blue Brain project, what was your role at that point in time? What is the stuff that you implemented over there or your addition?
So I really started as a baby, as a fledgling in the Blue Brain Project. I was one of its first PhD students, um, and and the focus of my work in the Blue Brain Project was to really try and identify uh, principles of organization in the biological brain. Um, so the Blue Brain Project started by trying to demonstrate that it is indeed possible by using sparse experimental data on how neurons and their synaptic connections are organized in a tiny part of the brain called the somatosensory cortex in rats. So the somatosensory cortex is the brain region that oversees or that processes information on how we touch, how we feel, how we perceive, um, any tactile information, right? So, for example, when you're touching a piece of silk and you're touching a piece of denim, there are obviously differences in their structure, right? But how is this encoded in the brain? And how does the brain know that you're touching a piece of silk, that you're touching a piece of denim, that you're touching a piece of sandpaper or whatever? How does the brain know that this is the case? So all this is overseen in the somatosensory cortex. So the goal of the Blue Brain Project, um, the initial goal was really to try and establish a framework, a pipeline, where through using sparse experimental data, it could actually be possible to use the sparse representation to build a very dense model of a part of the somatosensory cortex, a dense computational model, right? So this is called an inverse problem in physics because you're only sampling a very sparse data set and are actually coming up with ways to extrapolate to build computational models with this sparse data set and then integrate these computational models into a complex framework, which is like, let's say, a so-called microcircuit of neurons, the somatosensory cortex, and then try and understand how the structure of neurons, the shapes of their dendrites, their axons, their electrical properties, how they interact with other neurons, so on and so forth. So how does all this come together to shape network activity? So this has really been the primordial focus of the Blue Brain project in trying to come up with a way, a data-driven platform to link structure and function, and then to predict certain paradigms of network activity in terms of the function, like how does the somatosensory cortex work with a certain configuration of neurons, and what could happen if this configuration is lost, so on and so forth.
Right.
So this has been the primary mission of the Blue Brain project. So we started with modeling a tiny little crumb of the rat brain, a tiny little crumb of the rat somatosensory cortex. So the size of this crumb that we modeled is roughly comparable to a grain of sand or a pinhead, right? And in this very little piece of the brain, in this very tiny ecosystem of the brain, we already discovered that there are about 55 different neuron types, depending on the shapes of their axons and dendrites, kind of like the diversity of flora in the Amazonia, right? There are all kinds of trees, all kinds of shrubs, all kinds of bushes, and yet they all come together to shape the ecosystem, the functioning ecosystem of the Amazonia. And similarly, our goal was to try and understand how all these different neuron types and their synapses come together to shape the dynamics of this little crumb of the brain.
Right, right. Could you talk about your current research? And since you are a computational neuroscientist, would you like to unpack the role of artificial intelligence, machine learning in deciphering the language of the brain?
So my current research is in really trying to understand how the signaling chemicals called neuromodulators are organized in the brain, are organized in the mouse, monkey, and human brains, and to try and utilize this knowledge of organization across single and their synaptic connections and network activity to really try and use this knowledge, implement this knowledge into models of deep neural networks that could then tell us what is it about neuronal architectures in the mouse brain or the monkey brain or the human brain that enable them to learn, that enable them to process information and learn. So for the moment, I'm focused on the neocortex of the mouse and monkey and human brains, and to try and really understand how neuromodulators are organized, how these impact neuronal and synaptic function, and using these to really inform the next generation of neuromodulation aware deep neural networks, as I'd like to call them. So we recently wrote a paper describing a framework on how principles of neuromodulatory systems, how they're organized, their rules across neurons and synapses. So how could we use these to try and inform models of deep neural networks? So how could this possibly tell us, or could this give us an edge on how the brain is learning information? So this is still at a very nascent stage. Um, but I think there is a lot of promise in bringing together neuroscience and artificial intelligence, particularly models of deep neural networks, um, because in some sense the, the, the organization of the brain is not very far from how a deep neural network is organized. So there are hierarchies, there are structures. So there's flow of information from one level, from one layer to another, so on and so forth, right? So, but I think a lot of existing deep neural network architectures are quite abstract, and there is some renewed interest in the field now to try and slowly incorporate biological detail to try and understand what adding biological detail, for example, in terms of neuromodulatory function, how neuromodulators function across different neuron types. So what does this mean in terms of learning? What does this mean in terms of information processing in deep neural networks? So this is something that's slowly coming into light, and this is really the major focus of my current research in marrying neuroscience, especially the role of neuromodulators in the neocortex, and how this shot in the arm injection of biological detail into deep neural networks could help them learn better, could help them represent information in more efficient ways. So this is really what I'm looking into right now.
Lovely. So with this, what you have got to understand and the state of art with neuroscience, neurotech, what are the applications currently which is currently being used in healthcare, and what could be, what could we see in the next 5 or 10 years? Because there are, you mentioned these deep brain simulators which are being used for healthcare. Could you address that?
So I think there are two parts to this. So one is, of course, using technologies in neuroscience to try and understand, for example, brain dysfunction, or let's say even function, to try and come up with ways to alleviate humankind of um, the kind of suffering that the society undergoes as a result of brain disease, right? So we always talk about brain disease, um, but we don't really have a very good understanding of the underlying mechanisms that signal the onset of brain disorders, right? So, um, like for example, we talk about dementia or Alzheimer's or any other brain disorder. So dementia really is a spectrum of brain disorders. And at the very end, at the very extreme of the spectrum is Alzheimer's disease. But we still do not have any idea of what are the cellular and the synaptic components that are actually impacted during Alzheimer's disease, or what could it or what is it that's disrupted in the function of cells and synapses that could trigger the onset of dementia or even Alzheimer's disease. So all this is something we don't know, right? Um, so on the one hand, I think the current interface of neuroscience and deep neural networks could possibly try and help us understand what are the underlying disruptions in the neuronal and synaptic mechanisms that could possibly trigger the onset of some of these diseases. Of course, we cannot understand any of this without adding some more biological detail to existing models of deep networks, right? And on the other hand, I think progress in deep brain stimulation and all these very interesting, promising techniques could try and help alleviate some of the symptoms associated with chronic brain disorders, like deep brain stimulation in terms of epilepsy or Parkinson's disease. So these are therapeutic ways that are known to alleviate the sufferer of some of the the very drastic symptoms of these diseases, right? So on the one hand, I think the intersection of neuroscience and deep networks, or the intersection of neuroscience and computational models, could try and help us understand the underlying mechanisms of neurological disorders. And in trying to understand these mechanisms, that could then drive the development of a lot more specific, targeted therapeutic treatment that could possibly enable us to alleviate, as I said, some of these drastic symptoms. So I think it's a two-pronged approach, in my opinion. So first, in trying to understand the mechanisms, and then in trying to use this knowledge to implement the next generation, uh, a neural technology that could help alleviate these symptoms.
The brain, I think it's one of the most fantastic organ in our human body. Understanding it is, I mean, understanding it completely will open up things which is we can't even fathom at this point in time. There's a certain part of it which is super exciting, because you mentioned, there's so many things it could address to. It could address to healthcare. It could address to intelligent machines. But then there's that other side, the other side of the possibilities of us understanding the entire human brain. There's these dystopian movies like, the series Black Mirror and many other show a world of what could be possible when we are able to completely understand, scan and read a brain. That could a lot of negatives also which could open up with a technology like that. Do you think about that? Is there anything particular that kind of worries you about the advancement of and acceleration of neurotech, neuroscience?
I don't think I'm worried about the advancement or acceleration of neurotech, neuroscience, because that's what we need to understand the brain. But I think there's a lot of hype on like scanning and reading brains and things like that. Yeah, well, yes, brain-computer interface is a very promising technique, but I'm not sure if it will let us read a complete human brain, because uh, whenever we talk about the brain, it's always a snapshot, right? Because it's not like I mean your brain is a still image, but what your brain is, um, that's really the result of all the experiences you've had over development, throughout your upbringing, um, all these formative experiences, which are of course very specific, depending on an individual, really sculpt your brain. And I don't think there's any way to try and really say, okay, I mean, right now I'm going to scan your brain, I'm going to read your brain, and I'm going to download all this information, all these experiences that made your brain what it is over the past how many ever years, how many ever decades, right? So this is something that's a bit bizarre and a bit weird to me as a neuroscientist, because it's not like taking a picture through your latest DSLR and saying that, okay, this is your brain. But the brain is an extremely dynamic organ that's, I mean, that has been shaped through decades and decades of experiences that you've undergone as an individual, right? And I think there is no one standpoint to say that, okay, this is the brain. I mean, it's a constantly evolving part of the human body, that's very heavily influenced by our past experiences, and possibly uses these experiences to try and predict the future. And this is very dependent on the individual. So indeed, I'm very excited about prospects, about developments in neural technologies to try and understand, to probe the depths of the brain, to try and access all these parts of the brain that haven't been possible before. But on the other hand, to say that, okay, maybe I scan your brain, I read your brain, and I upload your brain digitally and things like that, to me, that sounds slightly far-fetched at this point in time. So I'm very excited about understanding the brain with current technologies, and all the more so to help alleviate brain disorders with this understanding. But anything beyond that, I think, would be a bit far-fetched, in my opinion.
Right, right. So I had the opportunity or the privilege to have a conversation with two neuroscientists who are working on the same. Uh, Not just scanning the structural and function capabilities of the human brain and eventually kind of map it to a computer. And the timelines, Kenneth Haworth I'm speaking about is maybe around 50 years is what they say. So yes, I think technology, it's a double-edged sword. It's got so many pros and cons. It is what that has shaped us as humanity, and it will always create a better future, because innovation is the only way for us to accelerate and grow as a mankind. But I think there is always fringe elements. It's not the technology per se, it's the humans who kind of direct it to a specific space, which could kind of go wrong. So yes, I'm not saying that this I have my personal opinion is that technology is ever growing, and maybe not now, maybe not 2022, but maybe 2080, 2090, this all could be possible. And I think it's needed for us, for people who have been vested in the space, to have a conversation of where this technology is growing, so we make sure that the technology shapes into a space where it's human first, and because that's the way it should always be. Now there is a lot of growth when it comes to the acceleration in technology. There's GPT-3, and the OpenAI CTO mentioned that GPT-3 is slightly becoming conscious. Joshua Bach, another AI engineer, is saying the same thing, that GPT-3, it's an NLP, natural language processing, and it's slightly getting conscious. Is it way too far-fetched as a computational neuroscientist? What are your views? Do you think that our understanding of the human brain could lead to thinking, intelligent, possibly conscious machines?
I think understanding of any brain could lead to the design of intelligent or conscious machines, perhaps. But the thing is, what does it mean by understanding the brain? Because as I said, it's not a snapshot, right? Okay, so let's take the example of the wiring diagram that I mentioned before. So a primitive worm like C. elegans, right? So which has about 310 neurons. So the wiring diagram in this primitive worm has been completely mapped out. So we know what neuron connects to what other. We have the whole blueprint of the wiring diagram of this primitive animal. And yet, we don't understand what this little brain does. So we're only talking about a primitive brain of 310 neurons. And the human brain has 86 billion neurons. Right? So, well, yes, perhaps these advances could help us understand something about the brain, but I don't know. I mean, okay, I'm not exactly into the GPT field, right? So my work is a lot more academic, and I'm a lot more interested in how the brain functions. Of course, if the outcomes from my work could influence the development of certain technologies, then very good. But on the other hand, I don't even know what it means when someone says, oh, GPT is like slightly conscious. So we don't know much about consciousness, right? Like people like Giulio Tononi or Christoph Koch or even Anil Seth, I mean, they've been looking into theories of consciousness and stuff like that. So there's this integrated information theory that Giulio Tononi and Christoph Koch have put forth. But we still, again, the brain is nothing if it cannot be understood in terms of its components, primarily neurons and their synaptic connections. So if we do not know what are the neuron types that are involved in making us conscious, what are the synaptic connections, and more importantly, what are these neuromodulators doing to make the brain conscious, then I don't think we are there yet. So to me, I'm a bit more grounded in terms of how I'd like to study the brain. So to me, understanding the components of the brain and what they do to make the brain whatever it is, to to enable information processing as a global network is very important. And I don't think we are there yet. And unless we try and understand the functions of these components, I think we are still a bit groping in the dark when we are already trying to attribute consciousness to machines. So to me, using what we understand from the brain, so extending this into computing architectures, artificial intelligence, machine learning is one way to try and understand the brain, but we don't know what these components in the brain are actually doing. So I think, yeah, I don't know. I don't know if 50 years is a realistic timeline, or maybe even 100 years. So we're not there yet. We definitely know a lot more than what we did like 50 years ago, but we're still not there yet. And I think it's going to take us a lot more time, especially with the human brain, given that there are a lot more technical difficulties to access a human brain tissue to try and study this directly. I think this is going to take a lot more time.
Fair enough, completely W. So, to my listeners, the ones who want to kind of understand computational neuroscience or want to take it up as a technology, as a career, would you kind of advise on some courses, content, or books that you can kind of recommend?
I think Coursera is an extremely good resource. There are tons of very good computational neuroscience courses. Like for example, I contributed significantly to two course era kind of modules. Although I think they are on a slightly different platform called edX, but nevertheless, it's all the same in terms of spirit and meaning. So Coursera is very good because it's really this massive online open access course platform that lets people learn um, through their own pace, right? So they can learn as and when they want, and this is really state of the art, and it doesn't cost them anything. So Coursera is an extremely good resource. Coursera or any MOOC platforms out there. Coursera is one. I think edX is another one. So utilizing these would be extremely good. And in general, just reaching out to people on social media and trying to engage in conversations is also another way to learn. I mean, books, yes, but books are bulky and expensive. So although they are useful, I'm not sure they are really the vogue right now. So I would really try and encourage people who are interested in learning a lot more about brain, especially Indian students, and India, given that it has such a huge technical manpower and people who could tap into all these online resources to try and translate their technical knowledge into something a little more applied. For example, in trying to understand the brain, I think these online resources like Coursera or edX MOOCs are extremely important. And there are also these online conferences like Neuromatch Academy, which is free, mostly free, and all virtual, all open. So people can really jump into the bandwagon, learn from experts in the field, interact with them, and really use this platform in helping them bridge their technical knowledge and apply this to something as arcane as trying to understand the brain. So all these are resources that I would urge people to try and make use of, especially students in India. We have like lots of people with the kind of technical know-how, with the technical background, and this whole new bandwagon at the interface of neuroscience and artificial intelligence is extremely alluring, all the more so for people in India. So if they could really ride this bandwagon, use these resources, and inform them a lot more about how the brain is structured and how it functions and how technology can be applied to understand the brain, then I think there's nothing like it.
Lovely. Thank you, Srikanth. Really, really appreciate you taking time and being part of the podcast. My last question to you. If you had to paint a picture of what the world is going to look like in the next possibly 10, 20 years when neuroscience, neurotech kind of completely, matures, that would be a wrong word to use. But once it starts, benefiting this thing, what do you think the world is going to look in the next couple of decades?
So I think what's important is to see how all this knowledge could help us alleviate brain disease. I'm trying to understand the mechanisms or rather the mechanisms underlying brain disease, and how we could come up with ways to actually treat brain disease. And to me, I don't know if the next 20 years is even a realistic timeline, but if we could even make a little surge, a bit of a leeway in trying to understand what are the underlying mechanisms of brain disorders, and how we could use this knowledge to come up with therapeutics. So we could somewhat try and alleviate the world of at least a bit of the sufferings that mankind has to go through in dealing with brain disorders, then I think that would be something very remarkable to me. So if we could try and understand, or if we could try and provide someone who's suffering from Alzheimer's or Parkinson's disease even a slightly more better quality of life, So, or if we could try and come up with ways to prolong the onset of these devastating diseases and really help people to lead as normal of a life as they've been leading before the onset of these diseases, then I think that's already something remarkable.
Lovely.
And I don't know if we can do this in the next 20 years, but there's always hope. We should keep trying.
Right. Yeah, I think it's the hope that we live with, and the growth of exponential technologies and the convergence of technology is something for which I'm super excited for. And I hope that these things which look a little far-fetched, I hope it comes a little closer, because a world is full of suffering, and if we can pull it down a little bit, the world would, have more smiles on, more faces. So I wish for a world like that. So really appreciate you taking time being part of the podcast. And to my listeners, if you like what you see in here, then please press the subscribe button. Until next time, see you guys. Bye-bye. Thank you, Srikanth. Really appreciate this.
Great. Yeah, thanks a lot for the chat. I enjoyed it thoroughly.